First of all, thank you for the invitation. I'm very happy to be selected to present here today. NurExone Biologic is the second time that we present here in this forum, and I have some news and update to give. We are a biotech company, a listed company at the Toronto Stock Exchange Venture, and the titles maybe gives everything, but we will explore it during our presentation. So where nature ends, NurExone begins. What we are doing is transforming the regenerative medicine. How we do it, you will find out in the coming minutes. So disclaimer, as a publicly traded company at the Toronto Stock Exchange, I need to have it. I'm sure that you are not going to read it fully, but let's continue. Our mission is to develop regenerative medicine specifically for spinal cord injury, and more specifically for the acute spinal cord injury. But I'm very happy to say that we see also effect on other indications, especially in the central nervous system. And what we do in 3 words is to regenerate the neurons, to rewire them, and to see functional recovery. How we do it? We have one product, our lead component, lead product called exopitan. It's not the only product that we have. You will see it in our portfolio, but this is the most advanced one, and this is show motor, sensoric, and functional recovery in several proof of concept that we made on rats. We are a preclinical company after pre-IND. The company was established just in 2021. 2 elite university joined together in order to show regeneration of the spinal cord. Professor Shalomit Levenberg from the Technion and also Danny Offen from Tel Aviv University had a project together. This project showed very strong results, which were published in 2019. And from there, we took it as a company. The company has 2 models. One, using the exotherapy platform for our product. We firstly and centrally focus on product production and getting it to the clinic. But because our IP, intellectual property, is so strong, this platform is universal, and this covers 3 elements: how to produce exosomes from 2D to 3D, how to load exosomes post-production after you get it in naives, and what to load inside. And we are specialized in siRNA delivery system. And the target, as I said, is acute spinal cord injury. We were moved to become a public trade company by June 2022. That was a reverse takeover from taking Inner Spark, a shell company in the field of mining. And now, after completing this transaction, we could buy that. And during this process, could raise up to now around $15 million for the company. And the structure is like that Canadian company, NurExone Biologic Inc., holds 100% of NurExone Biologic Ltd, located in Israel. And the R&D and the majority of the staff are in Israel. Recent development, and I think this is a good time to stop and say, okay, we achieved some tremendous achievement in the last year. First of all, we completed pre-IND meeting with the FDA. We got a very detailed information and guidance from the FDA, what to do and what not to do. One of the things that we should not do or recommended not to do is we don't need to go to large animals before getting to humans, which shorten our way to the clinic, shorten our way for first in human. And this is very important for NurExone. Second, last year we received the orphan drug designation by the FDA. And last month in November, we got the EMA orphan drug designation. So we have 2 continents with the same definition and the benefits that comes with that. This is exclusivity, market exclusivity, not patent exclusivity, tax benefits, shorten approved process for the regulatory path, and also the average price for that. Usually when you are getting to this status is around $100,000 per product. So small market, but very relatively expensive products. And what we're doing now, and it takes time, of course, is all the preclinical IND enabling study to tick all the boxes for the FDA and submit the IND, hopefully in the coming year. What are exosomes? I spoke right from the beginning, exosomes. But this is a new buzzword. The full definition is extracellular vesicles, is the small vesicles, small envelopes that release by the cells. On the left side, you can see the cells in the small envelope that released by the cells. And mainly they are for communication between cell to cells. They are not empty envelopes. They have inside cargo, which is RNA, DNA, several materials that helps to communicate between cell and cell, between healthy tissue to a damaged tissue, a tissue usually with inflammation. And this makes it as a guided missense. We use it as a drug delivery system with a lot of benefits. And exosomes is, as I said, as a buzzword, a lot of companies in the fields, and we see an advantage to use it compared to the traditional drug delivery system, which were chemically and artificial by human. What is the advantage about using exosomes, extracellular vesicles? First of all, you can have it in a minimal invasive way. I want to say loud and clear that our proof of concept that we made were done in intranasal administration. We deliver the exosomes, our therapy, through the nose. And from there, crossing the BBB, crossing the blood-brain barrier, entering to the brain, the exosomes find themselves where the inflammation is, where the injury was. And the injury was in the spinal cord injury, and it's targeted directly there. Second, the therapy itself, the exosomes, the envelopes, has a therapeutic effect. And so what we see is a synergistic effect between the delivery system and what we load inside. It's an off-the-shelf product. There is no cell. This is not personal medicine. One batch can serve several patients. That means low operational costs for the company. And the last one is that we are able to load external cargo, like we do with the siRNA that we use. How it looks schematically, we take the exosomes, we have our active pharmaceutical ingredient, API, which is in our case, siRNA, is a small sequence that help in the process of downregulate, reduce a certain protein. In our case, for the exopitin, we target the PTEN protein in order to promote proliferation and exon growth. And those are the results that we see. On the right side is the rat that after the treatment could walk again. And this shows functional recovery motor-wise and also sensory. If we are going into the tissue levels, in the middle, you can see that the neurons where there was a gap, which was a full transection model, you could see that the gap is closed and neurons get into it and rewire the spinal cord again, which allow the functional recovery. We repeat it again and we show the same results. DAT results show direction. What is the vector of the spinal cord, how the molecules of the water moves. And if it's green, that means it moves smoothly. If it's blue or other colors, there is a connectivity between the neurons. And you can see that only in the treated one in the middle shows very strong similarity to the intact. Intact means we didn't touch it. So this is how the spinal cord should look like. The middle is how we treat it, and you see regeneration of neurons, and there is no loss of neurons after the injury. But in the full transection, you can see a very significant loss of neurons. So if you summarize the advantages of exopitin compared to different methods, we tick all the boxes. That means that we can work in all the fields and answered the critical points that needed in order to develop the drugs. So we can use it off the shelf. There is no cells inside, no immunologic response. We can deliver it intranasal or intrathecal, and we show one of the only company in the world that show full transaction model, because it's like a kill experiment, or you have it or you don't have it. It's a 0-1 results, or you can show, if you can show regeneration, that's the model that will show it in the strongest way. So the product is protected 360 degrees with 5 families of patents. One of them is already granted in the US. By the way, not only in the US, also in Japan, Russia, Israel, and we're waiting for other territories to be allowed and granted. This is the umbrella patent, which covers the exopitan in the minimal invasive administration for regeneration of neurons. The other patents are all in PCT and in national phase already. The second one covers the production and the composition of matter of the exosomes, moving from 2D to 3D production, so large scale production. Third is what we load inside. This is the sequence and the modification of the sequence that it will work and create a synergistic effect. And the last one, and I think it's the most important one, because it's very universal and relevant for other companies, is how you load those exosomes, how you get your cargo inside the exosomes post-production. That's opened a huge door for us to collaborate with large pharma in order to be their answer for their drug delivery system. So take a product, use our exosomes, and then have a new product with maybe less toxicity or better efficacy that can cross the BBB. So that gives the right explanation why the exopitan is just the tip of the iceberg. Why? Because we show that it costs the BBB, and other product can do the same because it's the same envelope. We just change the cargo inside. It's very safe in a low dose. And the ability to load different API, different active pharmaceutical ingredients, not only siRNA, also mRNA, peptide, and small molecules can be beneficial for other companies in collaboration with NurExone. So if we sum up the market, first of all, we start as a spinal cord injury, and we will continue like that. It's a small indication, but as we said, we received the orphan drug designation, so we get all the benefit, regulatory benefit from this status. But we tried also the glaucoma model. The glaucoma model is a much larger indication. This is in ophthalmology. And what we use is to use the exopitan, the same drug, in optic nerve damage. Now, it's not spinal cord injury, it's optic nerve damage. And we presented and published it in July this year, the first results. And coming soon, we will have the full results of this study. This study was made in the third party in one of the largest hospitals in the Middle East, the Sheba Hospital in Israel. So we are not involved in the project except providing the exosomes. And we show a very strong efficacy and the ability to regenerate the nerves in the optic nerve. And what we saw and we shared is the ERG measurements after 18 days post-injury. And this show a very nice results. And then the third is, of course, the platform technology and collaboration with different companies. One of them is IntelliGex that we received this year, a grant from Eureka, and we work together as a combined therapy for chronic patients of spinal cord injury, combining cell therapy and exosomes together. That's exactly what I say here. So it's a 2-year grant funded by Eureka. And this is our answer together for chronic patients and get the best of two of the company for this very challenging indication. This is the roadmap for the company, tailor or focus specifically on the clinical study. We want to get the company as fast as possible for the clinical stage. It can be in glaucoma, it can be in spinal cord injury. In our plan, it should be very in parallel. It will run in parallel because the product is the same. It's a matter of formulation and matter of changing of administration, but the IND should be relatively equal and identical. This is the leadership of the team. I'm leading the team since 2021 as a CEO. I have a background in chemical engineering, physiology and pharmacology as a PhD from Munich, from the LMU Munich in Germany. Together with me is the CFO, Eran Ovadia, has more than 20 years experience with financing of biotech and also large corporate like Johnson & Johnson. He holds an MBA and also teaching in one of the university in Israel. The Dr. Noah Avnei, she leads the R&D. She is the director of R&D, coming from many years of experience in drug delivery system and also diagnostic. She had her PhD from the Nobel Prize winner or lecturer is Professor Aaron Shechanover, and Dr. Ina Sarel, with large experience with of spinal cord injury. He was involved with neuron in 2014. Hold a PhD in Boston University, and she covers all the aspects of CMC, quality, and regulation in the company. This is a scientific advisory board. It's a mixture between neurosurgery like Professor Nachshon Knoller, and ophthalmologist, Professor Michael Belkin, that made recently a huge exit with his company together with Alcon, the large corporate. Professor Gabriel Zelig, coming from the rehabilitation unit of the Shiva Hospital, and Professor Theodor Dagi. He is a neurosurgery and entrepreneur in the East Coast of the US. And he gives us the opportunity to already engage with some of the clinical centers in order to get as fast as possible into the clinical stage and have a very smooth start in a clinical stage. Director-wise, it's a combination between Canada and Israel. Together with me also today is Yoram Drucker, and he can also answer some questions here from the crowds. Dr. Gadi Riesenfeld was the president of Pharmos Israel, Pharmos actually US, and lived many years in the US, worked with several companies in NASDAQ. So we are very proud to have him on board. And we have the two Canadians on board. One nice story is that James Richardson, he was actually the CEO of the Shell company. He got in love with the technology and the people in the company, and we are very happy to have him on board also in our company, help us a lot in Canada to operate. That's it. I will open it for questions now. We have around 9 minutes. And a nice story is here. There's the first time that the open bell ceremony of the Toronto Exchange, or the TMX actually, was open outside of Canada and in Israel. And this case was open in Jerusalem, first time. That was July 2022. Thank you very much.
Thank you very much for the presentation. Lior, that was extremely interesting. Well, and more than interesting, we actually, very funny, we had a presentation earlier today by a medical technology company that was telling us about the problem of glaucoma and diabetes in the eye, and having actually no solution to some of the problems. So the fact that you're coming on your side in parallel with some proposed solution for that, which obviously won't be ready tomorrow morning, but bringing hope in that, I think that this is very interesting, and to focus on basically the hope that you're giving to a lot of people out there. We do have on one side, well, we have two futures colliding. One is obviously everything that you cannot do, you will be able to chip and replace and so on. The Neuralink basically kind of solution. And on your side, and on the other side, you've got your type of solution, which is actually regrowing, regenerating, and bringing back to a functional state. Can you tell us actually what do you see in the near future? Obviously, it's not ready tomorrow morning, as I said. I don't want to get people excited and thinking that this is a solution for right now. But how do you see those two paths in parallel, either fighting, collaborating, or how do you see the near future on that?
In general, I would say it's always collaboration, but I think I want to give a context for what we did in the last 3 years, and that will give you also you can extrapolate what will happen in the near future. We came from university, tech transfer, and one product, one company, one product, which was the XOP10. What maybe the name will stay the same, but it's now almost a completely different product, which allows us to be freedom to operate. But now, if you cut now, what is the situation in the company? We are a company with 3 products. The lead product is XOP10, and we have the platform technology. And then from there, you can take it to a different direction. And we could start to see it already this year, taking the same product with different indication. So if you can do it in a spinal cord injury, why not to do it in optic nerve injury? And why not to do it in other indication that requires a regeneration of neurons? So we will get deeper into the more indication, I believe. Second, the ability to have a very strong know-how how to produce exosomes and how to load exosomes is critical in this field. And I believe that we hold a very comprehensive solution for the industry in general. So I would say that we will see more and more collaboration with NurExone, and that will bring us also to a different maturation and different value and as a publicly traded company. And I think that is also reflecting our share price. Recently, we just published the EMA ODD, the European ODD, and that's the market reacts to that. And I think we will be able to produce more collaboration and results. And we are a very transparent company. Our average time for press release is a weekly. So we shared our results, and we're happy to have the investor, together with us.
Absolutely. By following your share price, we can see that actually the communication is surely at the center of it, because there are some fluctuation, but we can see some trends, we can see some movement and so on, and we see what I would consider kind of healthy trading and so on. Price never being stable, but moving up in up and down and with a very
And
clear
I
trend.
think in this case is also the liquidity. So you can see definitely that liquidity goes up. That's show maturation of the stock.
And discussing on that side, can you discuss a little more about the financial health and funding of the company? So basically, cash position, the last time it was published, and what is upcoming in the next 6 months?
We just finished one private placement in last 2 months, around 1.9 million Canadian dollars. So we have the cash to proceed with our, and execute our plans. We are publicly traded company. We are always get more investors during the development. Every step will bring a new evaluation, and each step will bring a new fundraising.
But basically, with that fundraise, you think that you're financed for how long?
So I present here several things. So if you are talking about spinal cord injury, we will have enough cash. If we want to go to the ophthalmology, that's require budget. If we have other opportunity, we will request, we will go to another fundraising. But it's very depend on how the company will develop, and I think the next coming months will be very interesting in my opinion, and that will, if we are going to establish the route to another IND, we will need to take it in consideration to have more investors and first of all collaboration, I would say first, because we as a small company with 18 employees, it's just one IND is very, very hard. So 2 IND, we need some collaboration and take it from there.
Perfect. And for the people that are a little less acute in the medical side, so what are the next steps of development between the result we saw and being able to apply it on a human, and what's the timeline?
Oh, that's a good question. So in order to get to a first in human, you need to complete the IND. We are around 1 year, 1 to 1.5 years away from IND. And that's mainly technical issues, not technical that it's not be able to solve. It's just better to tick the box for what is the spec of the product, how you control it, how you manufacture it. You need to document everything. We have already synopsis, which is what is our plan for the clinical study. This was mainly not, I would not say approved, but agreed by the FDA. This is also the material that you can see during the pre-IND. So the plan is there. There is a Gantt chart, and you need just to tick the box in order to submit it. And when you submit it, you have around 1 month to start the clinical trial. And the clinical trial in this case will be in the US, in Canada maybe, and in Israel. We want to have it in several centers in order to facilitate and the research and get the results as soon as possible, because the next milestone is getting into the phase 2.
Perfect. Well, this is great to hear. So we are out of time, Lior. I want to thank you very much for participating a second time at the IIF. I think that every time you come, it becomes clearer and clearer. I am personally absolutely mesmerized by the results you're achieving and what you can do. I don't think that, or I do not know if people realize how incredible it is that this is just a spray that you put to your nose. I mean, to me, this is absolutely fabulous. When I was a kid, dreaming about that would have been impossible. It would have been a big surgery or something else.
It's still like that. It's still like that. The only solution for a patient like that is surgery. So we try to offer a new route, and I'm calling all the participants to follow us on social media. We're very active, and be part of the story.
Absolutely. Well, we invite everyone, all attendees, to please follow your story. NurExone Biologic, you find them online. Go registered and receive their updated news. Every week you'll get it seems like an email from Lior, so or a press release. So keep up the good work. We're very happy to have you at the IIF, and best of luck for the next few months.
Thank you so much. Have a good evening.